From Microbe TV, this is Immune, episode number 42, recorded on February 10th, 2021. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back.
You have 8 feet of snow up there, is that right?
We got 2.5 feet. It was great skiing.
All right. From Durham, North Carolina, Steph Langle.
Hey there. Good to be back. Gosh, what's going on in Durham right now? I'm looking out my window. It's not particularly cold, but it's not particularly warm. It's kind of fine. It's good.
And from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. We also got more snow than I would like, but that's okay. Making it through.
Now, Brianne, I don't get it. You walk across like the street and you're worried about snow? Is that what I'm getting here?
I think it's because I grew up a little bit north of Cindy, and I think I got a lifetime supply growing up. And so I think I'm done.
Got it.
But
that's like Rich Condit. He spent many years in Buffalo, and they get a lot of snow up there, apparently.
They get a lot more snow than Ithaca.
We have a guest today. He is at the Rockefeller University, where he is a clinical scholar, Christian Gebler. Welcome to Immune.
Thank you so much. Thank you for having me.
Did I do justice to your name? Is that all right?
Perfect. Very well.
I'm not good with the non-Italian, non-Latino names. You know? Sometimes I can get it.
You got it.
All right. We actually had your collaborators on TWIV last week.
Yeah. I was wondering if there's something left I can tell you guys.
Oh, there's plenty. We actually didn't.
Oh gosh,
we didn't
so
touch that paper very much, but we anyway want to get your perspective since you actually did the work, whereas they didn't, right?
Oh, they did, and they did a great job. It was a great episode, and
yeah, it was fun. Before we talk about your paper and your science, why don't you give us a little bit of your background, where you're from, and where you were trained, and all that?
Sure. So I'm from Germany originally, and I'm a physician. So I trained in Germany. I went to med school in Berlin at the Charité University Hospital, where Christian Drosten, for example, now is the head of the Institute of Virology. And so I found it interesting because I listened to Sally Permer's episode the other day, and she mentioned that very few people identify, or very few physicians identify as researchers, and that this is a big problem. So I'm also sometimes struggling if I can now identify as a physician scientist already or not. But so just a background. So I was in med school in Berlin, and so my ties with Rockefeller actually go back 10 years now, because I joined Michel Nussenzweig's lab 2010, actually, as a medical student back then.
Oh, cool.
And that was an interesting time because back then it was kind of a transition in Michel Nussenzweig's lab as well, because some of those techniques that we still use, that we now use for our COVID research, so those methods to identify and look at B cells, single-sorted B cells, identify and study the immunoglobulin repertoire, were kind of pioneered back then. And so for me, it was a great time to join this lab, because just then Michel transitioned to work on HIV. And I remember that I joined the HIV group, which was back then was 4 people. And if you go to the lab now, I mean, over the last year we transitioned again, but basically the whole lab was working on HIV. So it was a very, very interesting time for me, especially as a student back then, to learn science. I had a great mentor, Florian Klein, who's now at the University of Cologne, who's also leading the Institute of Virology there. Who taught me science. So I very much related to Sally Permer's episode, because the focus on mentorship, how to keep physicians in science, or how to bring them back, I think was very important there. But again, I was there. We were working on HIV and methods of identifying antibodies against HIV, broadly neutralizing antibodies against HIV. But I was still a student, so I went back to Germany to finish school. I graduated, I think 2013, and then I I was on my way to become a clinician, basically. So I was working in the university hospital. I was doing my residency in infectious diseases, probably triggered by my research of HIV. So I was working there. I did my rotations. I was working for over 4 years, went through ER, ICU, all that very clinical path. And I had very little time to do science. But then after some time, there was always this flame that was burning, and I wanted to go back. And the way I did this was, how can I say what triggered it to make me return to New York? There's a couple of things. So first, when I was a student, I met my wife here in New York. My now wife here in New York, who was a PhD student at Columbia, actually. Vincent, she was she's a political scientist and was studying at Mailman School of Public Health. And so we always wanted to come back to New York. And at Rockefeller University, there's I think, fantastic program that again goes back to Sally Permer. What she said, it's called a clinical scholar program that is directed towards physicians who had some research experience, but maybe who work more clinically, and it really gives a structure for physicians to bring them back, to join them up with a more basic researcher. It's a very well-mentored program. It gives us protected time from clinical work, because I think that's the main reason to why physicians cannot go back so easily into science, because the clinical duties it's very easy to plan a physician on night shifts. There's always a bigger need, so it's very hard to find, I think, protected time, and this program really offers that. And so I was lucky to return to New York as a part of this clinical scholar program, to come back to Michelle Nussenzweig's lab. This time my wife came with me, and we brought our little son, who was 2 months back then. He's
Oh my gosh, wow.
he's a toddler now. He's keeping us on our toes as well. And so the whole last year was pretty intense. And so I was, and I guess we're going to talk a little bit more in detail about this now. It was another interesting time, because although I started then working on HIV again, more on HIV latency and the impact that those broadly neutralizing antibodies that we discovered back then might have on the HIV latent reservoir. But yeah, over the last year, we shifted to do and work on, like so many others, work on COVID-19.
How long do you anticipate being at Rockefeller? Do you know?
That's a very good question. I mean, the program initially was planned for 3 years. I'm in my last year here now. It's a difficult question. My wife is a diplomat, actually. She works for the permanent mission of Germany to the UN. So she was on the other side of working on COVID. She's a human rights officer, so she was very busy on that front. So we will likely go back to Germany. So for now, we plan to stay at least until next year, but let's see.
Sure.
I mean, you moved away from HIV, but I presume you want to get back to it at some point, right?
Yes, absolutely. And we were lucky in the sense that at Rockefeller, we have Rockefeller University Hospital, which is primarily a research hospital. So during the peak of the pandemic, I mean, we're right there. If you've been to Rockefeller, I mean, I guess most of you have been, but for the listeners, it's right in on the east side of Manhattan, and we have New York Presbyterian, Weill Cornell right next to us. We have MSK, so we've been tucked into this institutions that during the peak of the pandemic were at their capacities, and we as a research hospital were very privileged that or not only privileged, but also I feel we had the obligation to use that capacity that we have, and that freedom of not being in patient care to use that for research. And so although we are at the university mainly focused on COVID-19 research, was also into lockdown, we were allowed to keep up our HIV clinical trials that we were running. And so we did not enroll new people, but at least those were studies that have been running for several years. And we were very lucky that we could keep on having our study participants come in. And so we've been doing immunotherapy studies with broadly neutralizing antibodies against HIV. And so those studies have been going on. They're still going on for that matter. And so we're kind of doing both at the same time as good as we can right now.
Yeah. Wow. What a whirlwind your last year has been. I mean, not only raising a toddler during a shutdown, during a pandemic, but then, I think you can say you're a physician scientist. I mean, one, you
sounds like it.
Yeah. You have a first author Nature paper, and in addition to, really contributing to the work to understand SARS-CoV-2.
I feel a little bit more comfortable saying it now.
So speaking of first author, let's talk about this Nature paper, Evolution of Antibody Immunity to SARS-CoV-2. So you're the first author, but there are 11 asterisks people who it says equal contribution.
We should say co-author, first co-author.
And one of them is Melissa Cipolla, who took my virology course last year. I don't know if you know that, but
she told me.
Yeah, she said that taking the course really got her interested in doing this kind of work. So, and she's applying to PhD programs now. So that's cool. That's very nice.
Melissa was very crucial. It's true. I think this fact, and just when you look at the author list, right, and this whole project, Theodora and Paul talked a little bit about it on TWIV in the last episode.
For sure.
It was an enormous collaborative project, right? There's so many people, and that was kind of the most fascinating part of this whole process as well, right? How many people stepped up, how many people really threw their expertise in, how we were able to work together, not only at Rockefeller, but also with people at Caltech, with people at Mount Sinai, with people at Weill Cornell. It was really this incredible project, and it really is hard to determine these author lists there. And so I think and there was also even just today there was another paper that we were so lucky to bring out, and it all builds up on these on the research that we started in March last year, and so many people that worked on this, and it's just it's nice to see the results now, and we're really lucky to be able to contribute to this. And but there's so many people that are involved, so we have to recognize that.
Yeah, I don't know how you coordinate all this. It's just I've never worked with so many people in my life, so it's baffling how it would work. But I'm glad.
I just got off a meeting. We have a Wednesday call with Paul and Theodora, for example, where everyone comes together really to discuss the newest results and the directions. And those are fun. But it's definitely.
How many people are on those calls?
So this is a little bit more the core at Rockefeller. Okay. So between our virology and immunology labs, those are like 15 people. But then we have like you guys, we have Zooms every day, basically.
Yeah, right. So why don't you tell us what was the goal of this to start us off?
So maybe I have to start even a little bit earlier, because I feel like all our work is kind of connected. So it all started even basically a year from now. I just talked to my colleague Davide Robbiani, who's now he moved to Switzerland. He's leading the IRB, I think it's called, funny enough, IRB, so Institute of Research of Biomedicine in Switzerland. And he followed Antonio Lansavecchia there. But he started our clinical protocol here at Rockefeller to study people that recovered from COVID-19. And that was in end of January, early February last year, and I think none of us really understood what kind of scope that the whole pandemic would move into. But so early on we got IRB approval to bring people in who recovered from COVID-19, and so we had those protocols in place when we then finally understood. I think towards the end of February, early March, when New York went into lockdown. How crucial this was, and when we really started working on this project. And the good thing that we had this protocol in place is that we had everything ready. We just needed to find people, which in March wasn't really easy because everything was in lockdown. Logistics were very, very difficult. At some point, we even thought of putting together a mobile team to go into New Rochelle, which was this early cluster here in New York, to get maybe a blood drawn there. I mean, you all remember it was this whole, we didn't even know about what is the route of transmission? What do we need to do? It was really we were figuring it out as we went along. And but then at some point towards the end of March, we had then all the things in place, and we were able to reach out to individuals, and we were able to bring them into Rockefeller University Hospital for a blood draw that would then allow us to study their immune response. And so from April into May, we brought, I think it was in a time span of 6 weeks, we brought in over 150 people for a single blood draw. And based on that, the meanwhile was used by Paul and Theodora, for example, to set up those neutralization assays. We used those techniques that were pioneered and used for HIV first in Michel Nussenzweig's lab to study the immunoglobulin repertoire. And that was kind of the first initial study of the immune response in those convalescent individuals. And the goal was then to, I think when we went through the summer and early fall, I think one of the big questions that was on everyone's mind was how durable is the immune response, and how does it look like? And so we went back. We were so lucky to have a very committed group of people, one must say. And so over around 100 of them came back to follow up with another blood draw. And then we started basically using the same techniques and trying to understand how the immune response after 6 months looked like.
So when you were looking at that group of people, which it's so fantastic that you were able to get those patients, how representative did you find them to be of all the COVID cases that were being seen?
Yeah, that's a good point. And we also understood that then early in April quickly, because obviously the individuals that were able to come into the hospital were not the individuals that were in the hospital for a very long time. So our cohort is certainly more on the mild to moderate symptomatic side. We tried to change this a little bit and enroll some individuals that were also hospitalized. But in general, it's true that this group of people is a cohort that had mild to moderate symptoms, so that one has to take that into account. And I think that goes back actually to that point, because I think when we started trying to understand how durable the immune response is, I think there were several studies that came out that kind of questioned or would suggest that the immune response or the durability is not very long. And I guess that was mainly driven by studies that were potentially in individuals that were more severely sick, that maybe had already not the strongest immune response in the first place. So we wanted to take a look ourselves, and we were lucky to bring those close to 100 individuals back.
So you, having worked in HIV/AIDS, I wonder, my understanding is that people who participate in clinical trials are really into it. They're very happy to participate, and really they want to help each other. Is it the same with the COVID patients? Do you know?
Yeah, they're fantastic. I mean, I'm so thankful. We just actually today I saw 2 of them, because we're now coming close to the 1-year time point. So we're starting to bring them back for the 1-year time point, and they're still so committed. It's really fantastic. I mean, I kind of understand that in March, April, right, everyone was so eager to help. Everyone wanted to help, but even now, it's really a fantastic group. And sometimes we have to brace them a little bit and say, you don't have to come in tomorrow. You can come in. You can come in March. It's totally fine.
That's great. So looking at your paper, it really kind of divides itself into 4 different sections. You break it down by looking at the general plasma antibody response, then looking at memory B cells, followed by the monoclonals, and then antigen persistence, particularly in the intestine, which I'm really just kind of obsessed with that topic. So I'm sure we'll get to talk about that. But just looking at the plasma responses in general, what are some of the trends that you noticed?
Yeah, so I think, and again, it all goes back to the participants. We were so lucky to get those close to 100. So I think it's for those things, it's always good if you have a higher N. Of course, it could always be bigger if you do zero surveys, but I think for our purposes, we were happy to get at least close to 100 individuals where we could then really look at the activity. And what we saw is, and when we break it down by antibody isotypes, we did see quite a decline in the in binding levels, so measured by ELISA, but also in neutralizing activity levels. And what we saw is that, especially for the neutralizing activities, they were also proportional to the initial levels. So someone who would have a higher level to start with would also drop more. But in general, in most of the individuals, we still detected antibody levels, and it's not nothing that we would expect to see a drop in antibody levels. So I think the next part that you already mentioned, so to really look at memory B cell responses, and then go even deeper and look at the immunoglobulin repertoire, that's kind of then even the more interesting question, and what we followed up with.
So one of the things that I think is interesting in this set of data is the differences you see in the different isotypes. Mm-hmm. Was that something that you expected to see, or is there much known about different persistence of IgA versus IgG? Because that wasn't something I was aware of, but I don't think about antibodies as much.
Yeah, I mean, as you know, I mean, the steeper drop in IgM is very expected. So to see class switching into IgG, I think that's something that we totally would have expected. With the IgA, I agree just to recap, so we saw a lesser decline in IgA levels than we would see in IgG levels. It could potentially go back to the fact, when we talk about this a little bit later, to the antigen persistence, or maybe a more triggered immune response through maybe also different routes of infection. I mean, some other groups have seen other results, so they would see the same declines in IgA. So again, Brian, like you said, also then it comes back to how comparable are the cohorts. Is there maybe a difference there? It's difficult to say, but we did see this a difference in the decline of the IgA levels.
And it's interesting with IgA because we know that, there's a huge preponderance of them in the mucosal surfaces. So you could expect, if there is some type of mucosal stimulation going on, that you would have persistence of IgA. But what's interesting to think about is, you have monomeric secreting IgA cells, and then you have dimeric, which would then end up in the mucosa. And so what really are those antibodies? Because I'm assuming the secondary was just looking at all IgA, correct?
Yes, that's correct. Okay. But
yeah,
actually, we have another paper that specifically looks at that. It was in Science Translational Medicine. I think it was published sometime in the fall as well, where we actually then produced or looked at the repertoire, and we looked at monomeric IgAs compared to dimeric IgAs, and we did see that the potency of those dimeric IgAs is vastly increased due to avidity effects. And an interesting part of this paper as well, because it's the same cohort that we looked at, that I found interesting that we didn't really discuss now in this Nature publication, is that we saw a good difference between individuals that had gastrointestinal symptoms during acute infection and higher IgA levels. So that was an interesting finding that we had there.
Yeah, I remember that dimeric IgA paper. Love that paper. But it was interesting when I thought about that paper in regards to, so if your neutralization data in general decreased over the 6 month, but the IgA stayed the same, it may suggest that maybe it's not represented by the dimeric producing population, just because the NEUT data doesn't correspond to that. But I'm sure you're looking into all that. We always joke with the grad students and postdocs in our lab, like whatever hypothesis you have, you just assume the Nuisance YLAF has already thought of it. They have funding, they have data, and they're going to publish on it in like a week. So it's kind of a running joke with us.
That's probably true.
Could I ask you, you looked for RBD and N antibodies, right? And
correct.
So why did you... explain for our listeners why RBD alone and not total spike, and why N?
So it's a good question. I mean, in the initial paper, we also did total S ELISAs and only RBD. I mean, we know that the receptor binding domain is the most vulnerable target of this virus. So maybe those are more meaningful when we measure the titers. And additionally, when we do our next steps, we use a fluorescently labeled RBD and not a spike protein to do the single cell sorts. So I think those are just then more interesting for our ELISAs and to do the plasma activity as well. For the N, yeah, it's that was another thing we did analyze us. We also used a commercial assay to look for N, and in this case we actually saw an increase in the levels, although that might have been more an issue of which was a lateral flow assay, not an ELISA. So maybe that was more of an methodological thing that we saw there. But there are actually several papers that use the same. It's the Roche assay that is a lateral flow where you actually see increases over time. But I think that might be more inherently to this assay.
I was curious, since you've been able to follow up these patients, or you're starting to follow them up, do you have a cohort that's been reinfected? And can you do any sort of comparison to find out how their initial response compared and whether or not they're more susceptible? And can that tell us information about how this whole thing is going to evolve over the next year or so?
Yeah, Steph, the Nussenzweig lab has thought of this. So Cindy, yeah, that's true. We're actually doing this right now. We're doing this right now. We're calling them back right now. I know of one individual who was reinfected in our cohort. We will see. I guess we will also find out, or we might have some asymptomatic contacts then, right? I think we might find out by looking at their antibody levels, right? If we see, we would expect probably very, very low levels at this point. But if we find someone who really spikes in the antibody levels, that might speak to someone. Let's see if they have a history. Maybe they had like another COVID scare or not, but we might see those even just looking at the antibody levels.
Yeah.
Within the first set of results, you had talked also about patients who had these persistent symptoms, and you had listed what that meant. You had to have some type of fever or recurrent cough with 3 other symptoms. Can you talk about what those results were? And would you categorize these people as having the long COVID? Is that what you're trying to profile here?
Yeah, I think so. These were things we even did quite early on after they came back. So that was in October, and I think I'm still not sure if we have a really good class. So this is kind of like an in-house classification that we tried to come up with. I don't know if we have a good standardized definition for now that you can say this is long COVID or not. But for us, it was pretty striking, and we took our time at the 6-month follow-up to really we had like a standardized questionnaire for our participants, because we really wanted to capture the time or what happened between the first time we saw them and until the 6-month time point. And so we were surprised to see how many would still mention that they had some at least reduced physical or strength or some fatigue symptoms. A lot of them still had anosmia.
Wow. And
back to your question, Steph, so we kind of, we didn't even count anosmia as an individual symptom for persistent symptoms. We kind of made this like because we would have had so many of them. But still, when we even had the more stringent criteria, even more than 40% of our group, which again was also mild to moderate, had some form of persistent symptoms. And when we looked at the antibody levels there, we also did find a difference that individuals who would report persistent symptoms had higher IgG levels, but they also had stronger symptom or more severe symptoms at the acute infection. So that might just be a function of that.
As far as you know, these long, or whatever you want to call them, they're not infected. They're PCR negative. Is that correct?
Yes, they were all PCR negative. Mm-hmm. Everyone was PCR negative when they came.
By swabs, not stool.
We actually do saliva.
Saliva, right?
We have a Bob Darnell at Rockefeller developed a saliva-based test, so they were all saliva negative.
So do we know about stool shedding? Just trying to correlate what you were saying before about antigen stimulation.
We did not look into this. Mr. Rappenhunter, who was our collaborator at Mount Sinai for the intestinal biopsies.
Oh, right. Okay.
They came in with a nasopharyngeal swab, but we did not look into stool shedding.
Okay.
So is it in general the idea that those individuals with more severe symptoms have higher serum antibody titers?
Yeah, I think you can say that now that symptoms do correlate with, or severity of symptoms does correlate with antibody titers. It's not a very, very strong correlation, but you do see it.
And that's
And
probably
when you look at hospitalized individuals compared to outpatients, they usually is a clear signal.
But I guess that's an immune question, chicken and egg, right? So
Yeah,
is it because they have more virus that they're generating a more robust response and their antibodies are higher? Or is it actually that they're, even to the same level of virus, generating a more robust response that's then responsible for immunopathology and symptomatic disease? We don't have any insight to that yet, do we? Do you have any thoughts on that?
Yeah, no, I agree. It's
Yes.
I don't know. For this
Yeah.
one, I don't.
I guess it would require a kind of study a longitudinal study where you sample people
Yeah.
frequently over time, right? And then hope some of them get serious disease and some of them don't and see what the difference is, right? That's about all you can do.
Yeah.
Yeah, it's super interesting. Huh.
You almost have to have samples before they were infected as well.
Yeah, kind of tough. Huh?
Well, there have been studies where they've been able to go back and, do samples that were banked, but it's not quite the same. Right.
Right. Well, so I think in general, looking at the plasma response, you in general you saw that decrease over time, or over the 6 months.
Yes.
So then when you move to, and you can talk about whichever first, but if you want to talk about functional antibodies or the B cells, you start to see maybe some differences.
Yeah, correct. So the way we look at the plasma. Our participants come in for the blood draw. So we look at the plasma, but we also have the cellular side. And so what we do here, and these methods that have been established in this lab, so we isolate B cells and then we try to look for SARS-CoV-2 specific B cells. And the way we do this is by taking a fluorescently labeled RBD and basically use this as bait to go fishing and then try to fish out those SARS-CoV-2 specific B cells out of the whole pool of B cells from the individuals. And I'm always very impressed by these machines. I still look at them. I mean, they're bulky. They look like enormous machines, but they're elegant in a way that they can really sort individual B cells. And this is still to today very fascinating to me, that you put them into 96 well plates and there's an individual B cells. And so that's how we do this. So we really use the RBD and then we try to fish those out. And once we have them isolated in this one well, we basically can use PCR amplification techniques to recover the immunoglobulin heavy and light chain genes, do the sequence those, do sequence analysis, and that already gives us a good idea. But then we can also basically clone those genes, put them into expression vectors, and produce those antibodies. And Melissa is a master at this, Vincent. So she's been helping with the especially the cloning and the producing of those antibodies. And what this allows us is to then not only be on a bioinformatic level to look at the sequences, but really put those antibodies to test and see what they're doing.
What is the number of RBD specific B cells in a typical patient?
So they're tiny, right? So when we look at the fraction of those from the pool of memory B cells or IgG-specific memory B cells, they're very tiny. So usually between 0.001, something like that. So sometimes for some individuals, you really just get 196 well plates because you can only find 96 of those cells. For some individuals, it's much higher. And then sometimes it takes a long time, but it's not easy to find them. But once we have them, we can study them. And what we then also see is, that's the fascinating part of this about these machines. Again, they're very good at it. So those actually, when we produce those antibodies, they recognize SARS-CoV-2. And sometimes we take that for granted, but I'm always surprised when we don't see this antibody is actually neutralizing SARS-CoV-2. So this machine did a good job.
So there's like real benefits to that because you can isolate those cells, you can clone the antibodies, you can express them, we could use them for various things. But can you talk about what are the limitations? Because Vincent has a good point. You know, there's very few of these in the blood, and we know that some of these B cells, especially memory B cells, hang out in other places other than the blood. So can you comment on like what is the representative pool that you think you're actually looking at when you look at peripheral blood? I realize the limitations, you can't go just taking people's lymph nodes and gut and everything out to try and locate these things and do bone marrow, aspirates and things. But we sample the blood, but what does the blood actually represent as far as the total B cell pool?
Yeah, that's a good question. I mean, for a lot of questions, it would be fantastic, right, to get lymph node biopsies, look at early germinal center reactions, all those things. It would be really great. Or like local reactions like gut biopsies and really do those studies. Yeah, you're right. I mean, we're looking at the periphery, and it's so many studies and so many methods that we use are based on that, because it's just the simplest way of getting those samples, right? But I think it is pretty representative, because also one of the findings that we then had, right, is that when we look at different individuals, we find those overlapping, almost identical antibodies and memory B cell with the same memory B cell receptors, what people now start referring to as those public clonotypes. And so I think it is representative, but of course you're right. I mean, probably we might see different things, and even scientifically, even more interesting things if we go really to the source where they're expanding, where they're maturing, and that I'm not sure if our participants would be so eager to come back for those studies.
You're like, how committed to COVID research are you guys?
Right.
So thinking about, the results and for our audience, what we just described in the plasma is really representative of active stimulation, active antibody production. It's kind of like that acute response in terms of adaptive immunity, but it's going to wane, because if it doesn't wane, you're going to have this production of antibodies that you don't need. And that could lead to then binding to things like your own self. So you need those to decrease. And that's why you saw neutralization decrease. But with the memory B cells, you do see an increase in the number of nucleotide mutations. Somatic hypermutation was very variable amongst individuals. And so what does that tell us about memory and about the future if somebody were to get reinfected?
Right. So that was then the next step. And that's, I think, where it was getting interesting, because you heard people say what we saw is that memory B cells actually stayed stable or increased in contrast to the antibody levels. People said, well, the immune system is doing what it does. That's what it does. And this is true. This is true for HIV. We've been seeing this, that HIV is a chronic infection. And especially for SARS-CoV-2, again, we had those initial studies where people really questioned the durability, and people really questioned even germinal center formation, right? We have this study from Shiv Pillai's group, which looked at germinal centers and the lack thereof. And so people were really questioning this. But when we then saw that, when we looked at the repertoire, when we looked at memory B cells, we saw that the numbers stayed the same. And when we looked at, as you mentioned, somatic hypermutation, so we saw this maturation process in the memory B cell compartment that even made those B cell receptors give them a higher affinity to SARS-CoV-2, right? So we saw that our immune system was progressing in a way that the germinal centers were well and intact in those individuals.
So you mentioned that this is the immune system doing what the immune system does, and it's similar to what you've seen in HIV data. Is it also similar to things that you'd seen in acute infections, or is it really what you'd seen in a chronic infection?
I mean, HIV is obviously always like a somewhat outlier. I mean, we all know that HIV is a completely different beast, even when we compare it with SARS-CoV-2. But I think it's hard to tell now, actually, and we asked ourselves that too. How does this now, how do those findings compare to other viral infections, right? Unlike HIV, for example. I think we just didn't look at those, right? I mean, it's just we were lacking a lot of those. Like no one would have looked at those methods for a coronavirus, right? So I think we just don't know. Maybe that's one of the silver linings. Maybe that we also gained some basic immunological, virological insight in those questions that might help us to understand other viral pathogens better, right? So I really don't know how this compares. We really don't know.
That has been my take-home message over the past year as well, of all of the basic immunology and virology that hopefully we'll learn.
Yes.
Yeah, definitely. I mean, you're right. They say, oh, well, this is the immune response doing what it does. But that is really taking for granted, I think, what we haven't been able to show in humans, because why would you do this study? You do it in animal models. This is what it looks like. But it's really phenomenal, I think, to be able to see that. And in regards to that, the germinal center question, we discussed that paper.
Yeah, we did.
But I think the difference would be that those were patients who had succumbed to infection.
Yes.
So that was severe disease, correct? So and you only had the mild. So for some reason, their immune response did not allow, I mean, obviously an immune pathology-based disease, then the germinal centers really suffered from that.
Absolutely. And I think it's not contradicting anything. It's
Right.
exactly that. It's just a different group of people.
Totally.
So Shane Karate has, I think, done some similar work on antibodies and memory B cells, and from what I understand, he does see a decline in memory B cells, but he believes he can model out that these will last for years. Is that something you can do or you agree with?
Actually, Shane Karate also saw an increase in memory B cells. He saw the plasma activity that dropped, but he also saw stable levels and even a slight increase there. So I think that was very much in line.
And so he concluded that these could last for years. Is that a reasonable conclusion?
Yeah, I talked to our participant today, and he asked me the same question. What do you expect? I mean, we will see.
Is there any reason to believe that vaccine responses would be any different?
Yeah, that's actually the Nussenzweig lab has thought of this and just published a paper.
Yeah, today it came out. I'm telling you, whatever question you have in this field, they're on it.
So it's actually published today, and it's I think that's, so you can read many things in this paper, but the biggest message that I get from this is what a great achievement those vaccines are, because they resemble natural infection in a way that is amazing. Because when you even look at the immunoglobulin repertoire, those vaccines basically they just elicit the same antibodies that natural infection would do. And so we just this has been published today, and I think it's that's one of the key take-home messages of this publication, is that it's really amazing how good they simulate natural infection.
Yeah.
And I think one of the things we need to think about too is continued tickling of the immune response, because this thing is still circulating, right? And so it may be different once we actually get a hold of this and people are not being potentially exposed to it repeatedly and boosting their immunity potentially. So
yeah, it seems to me that based on the kinetics of what you're seeing, that you do see a decrease over 6 months, and what that threshold is for reinfection, I think we'll learn. Right. But you would likely then need that booster response of a yearly or maybe every other year, because otherwise it still takes time for the immune response to respond, correct? So you'd still have to wait 7 days-ish. So really what you have circulating in your blood at the time of infection matters. So I think it's really interesting to pair what you've done and the paper you just published to try to identify, okay, the kinetics of everything. And then when should you boost?
Yeah, it's a good question, right? Is it protect? It's again, like, is it sterilizing immunity? Is it protective immunity? It will take some time, although those memory B cells might still be there. It will take some time to create secondary germinal centers, to get plasmablasts again, to boost up those levels. Will that lead to reinfection or asymptomatic infection? I mean, my guess with protective immunity, I think is not unreasonable to believe that this will happen. But for transmission infection, it's hard to say. It's going to come down to the dynamics of the whole process.
Yeah, for sure. Great. So then, the next section of your paper, you did pull out those, you made monoclonals, and then you tested them against different variants, or at least I shouldn't say variants because the variants contain actually multiple mutations, but viruses that introduce different mutations that are relevant for the current circulating variants. Can you talk about that?
Yeah, and this was so we did clone and produce those antibodies, those monoclonal antibodies, and then we tested them against the wild type. And this was also October. We wanted to start doing those against the variants, but we did not know of those now circulating variants of concern. So what and Paul and Theodora did and their lab did is they already looked at some that were actually like point mutations that were circulating, and also based on BSV selection screens and some mutations that would possibly come up, and start looking at those. So they created those RBD mutants, and also with Pamela Bjorkman at Caltech together, they created those mutants that we could then test. And what we've seen is that those matured antibodies, so we saw that the levels of somatic hypermutation for all individuals that we looked at increased significantly. And then what we saw is that those matured antibodies were actually then had higher affinity to wild type, but also were able to bind to RBD mutants, and also in a functional neutralization assay then as well. So we would compare B cell clones and their monoclonal antibodies from 1 month after the infection, and compare them to their children antibodies, if you might say so, that matured, and they would then generally be higher affinity, but also be able to neutralize some of those RBD mutants, which was interesting to see.
Yeah, because essentially with the maturation of the antibody response is why you're able to have that breadth. But
correct.
With the vaccines, and this is probably going to touch on the data that's presented in the other paper, which I've read in a preprint, but haven't read the published one. Some of those some of those point mutations that are in the variants, those responses were lower. But what you're seeing is they're actually higher over 6 months. And so this is just the difference in the type of response you're inducing in natural infection versus the vaccines, or something else?
Again, so when you look at those vaccine plasmids, they were more or less taken more like 1 month to 3 months after, right? So it's an important question if there will be the same maturation processes that we saw for natural infection, if we see those for people who got the vaccine.
And maybe it's too early. Maybe you have to wait a little bit.
We will see. Or we actually did not do we did not compare after 1 month to 6 months, right? I think those individuals we will need to bring back and look at this, because it's an interesting question if those same maturation processes, if those depend on the full viral infection, or if you can recapitulate those with just the spike protein that you get through the vaccine. It's an interesting question. And I think it then maybe leads also into the last part of this paper. We were asking ourselves as well, like, is this something that we would expect, this type of affinity maturation, or what leads to these to this maturation process? And the way we did this is we worked with Saurabh Mehandwar. There was a collaboration. He was a gastroenterologist and scientist at Mount Sinai. And I think that came more out of an informal conversation with him, because he had the observation that once the healthcare system started seeing or having regular procedures again, that if he had individuals who would come in for endoscopies and who were recovered from COVID-19, you required a negative COVID test, right, to enter the hospital. So they were all PCR negative. Huh. But when he took gastrointestinal biopsies for other reasons, he also looked at some of them in his lab, and he did a staining for nucleoproteins, for SARS-CoV-2 nucleoproteins. And what he saw is that he actually could detect in some of those individuals, even 3 to 4 months after the infection, he would still see some viral proteins in the gut epithelia. And so that was just an interesting observation that might as well help explain why when you have those persistent viral fragments, that might explain such maturation processes. Mm-hmm. Although one must also say that it's, even if you would not have persistent viral fragments, the germinal centers can be pretty long-lived, and you could still have follicular dendritic cells in primary germinal centers and that is known also that still present antigen from the initial infection that would probably also be enough to drive such maturation processes. It was just an interesting observation that could help explain those further.
So go ahead.
I was going to say, I love the way you phrased some of this about persisting antigen fragments.
Yeah.
Yeah, great.
And so do you think that there is active viral replication, or do you think it is just persisting viral fragments?
Yeah, so maybe you're also, so in our first version of this paper, in our preprint version, we also took some of those intestinal biopsies and we sent it to Pamela Bjorkman and Mark Lodinsky at Caltech, who are more on the microscopy side and electron tomography side.
Yeah, what happened to those pictures? I looked at that paper. I'm like, what? They're gone. They got dropped off.
This is called peer review study.
I figured you might have some insight. I didn't know if you could share with us exactly what the situation was, but I mean, people can go see those, and it looks like a coronavirus to me. But
yeah, I have it here open, so I'm going to look for those. Yeah,
sure. Screenshot.
Yeah, so we have so just to explain, so we sent those biopsy samples to our collaborators at Caltech, and they looked with EM, and I was so there were some structures that looks like a coronavirus to me as well, and we also so this is why we also put this in our first publication, but I think. And I'm not even being sarcastic about this. I think this was very important, and this is why we have peer review, right? This is why we go through this, and this is why we during the peer review process this was looked at. And I'm not a microscopist, I'm not a structural biologist, so this was looked at, and it's also true that many things can look like viral particles. I think with the
for sure.
And so I think this was just not conclusively proven, and I think we just have to work on this. And Pamela Bjorkman, Mark Lodinsky, they're all very good experts at this. And we even had some videos. We were able to take some videos, and I think it was very convincing. One question was also to do some gold-labeled immunostains.
Yeah, that's what I was going to ask.
Which is not easy to do on those samples, but actually we were also able to do and identify some of them. But it was just not conclusive, and we just need to get new samples and work on this. But I think it's we will see.
Steph, is there another coronavirus that infects the GI tract of humans?
So where is the main tropism? No, but of other animals, there definitely are coronaviruses that are can ravage the intestine. That's their main tissue tropism. But what's interesting about coronaviruses, right, is you do see this. Maybe it's not the main organ where you see active replication, but you see shedding. So it can't just be ACE2. There's other things going on, although the intestinal enterocytes express ACE2. So this is a whole, whatever's going on in the gut, there's a lot to learn there.
I mean, I think Christian is right. You have to show it's SARS-CoV-2 particles. If you publish a paper with particles, then people get all excited. And I remember there were some papers published last year by clinicians where they did EMs of endothelial cells, and they say, "Oh, look, particles," but they're not particles. But everyone got excited and said it's replicating in endothelial cells, but you have to be careful. So I agree.
That's a good
that.
point. Do we know about the common cold coronas? Do they end up in the gut?
I bet we don't.
No one looked. I mean, there's actually one interesting study when we dig a little bit deeper from the 90s, working on feline coronaviruses, and that was actually there. They had this animal models in cats, and they did find they did some immunostaining. So that was not EM and not particles, but they also found in the epithelia, like I think after 6 months in those cats, they did find some viral fragments. So I think it's again a question probably no one has ever looked, maybe with some exceptions in those animal models. But even so, let's assume even if those are viral particles, to go back to your question, Brian, I'm not sure how, if the question is this replication competent. I mean, in the just the histological assessment, there was no active inflammation, which kind of speaks against active replication there. I think it's, and that's why I also I think just seeing those viral particles is fine, because I think it's more important from this immunological standpoint, right? That it's driving some of those maybe still ongoing germinal center reactions. And it's more interesting from this immunological. I'm not sure how and how you even prove that, even if you see a particle there, if that's replication competent or not. I think that's very, very difficult.
I guess the thought from that is that, how would these viral fragments remain around for that long? Right?
If they're not replicating.
We chew up everything, don't we? They would have to be very highly resistant to degradation or protected by some other mechanism. And so otherwise, it's hard to imagine. I'm not saying there is replication competent virus, Brianne. I'm just questioning, how could we retain those fragments around for 6 months or for a very long period of time? I would imagine they would have been long gone.
Yeah, and to me, the reason why this is the question becomes sort of interesting is the question of chronic antigen stimulation versus acute antigen stimulation, and how that's impacting some of these immune responses that we're talking about.
Right.
Right, and what generates an antibody? I mean, we know the rules of antigen presentation and the length of the peptides that are presented, but it goes back to what you said, Brianne, chronic versus acute. How much is there? And is a little bit, is that enough to stimulate B cell production? I'm sorry, antibody production from B cells. But those follicular dendritic cells, I mean, those seem to be a key cell type that you could look into to try to understand this question on a mechanistic.
Christian, what was the frequency of finding in your cohort? Or I guess the gastroenterologist had a separate group of people, right? So, I mean, was it every patient? Was it, or you can't tell?
No, it was about, we had samples from 14 people, and it was about in half. So it was also not in everyone. Mm-hmm. So it's an interesting question, but I think it needs, you need to look further in those individuals and try to answer these.
Yeah, I asked Paul on Friday, and I didn't get a clear answer. Maybe you know. So what about, is there infectious virus in the intestine, or is it, I think it's rarely been identified, right, in feces?
Yeah, I mean, even if you do culture studies, right? I mean, there's some actually from Christian Drassen's team in Berlin. I think they looked after, but there was also more acute infection. I think it's after 14 days they couldn't culture anything. So there are very, very few studies. I think it's just difficult.
So looking then, it'll be really interesting to see your 1 year. So the people that are coming back for their 1 year appointments to see what the durability is. Now, these individuals, they can't be precluded from getting the vaccine, but of course they could have gotten the vaccine, correct?
Yes, and some of them did.
Some of them did. So then you can look at the difference and I just saw, I was looking at a paper that the second vaccine in an individual who had been infected looked like the response of somebody who had not been infected and gotten vaccinated. So saying that there's a boosting response there.
Yes, I think we've seen 2 or 3 of them now. One from Fuller and Kramer, who looked at the antibody levels after dose, and I think they're sky high after the if you're convalescent and you get one shot, you're really boosting the memory that you had from before.
Right, and I know there was the implication that if you were infected within the last 3 months, there were people, I guess physicians maybe telling their patients don't get the vaccine. It might stunt the immune development. But I mean, obviously from a logistical implication, we just need people vaccinated. But there is an immunological question that is interesting there, is if you do get vaccinated within that window, maybe waiting is beneficial for your immune response, not on a public health level, but just scientifically.
Or the opposite, right? I mean, you could even think you have antibodies, you get some spike protein produced through the vaccine, you build immunocomplexes, you might boost it, right? I think this is a very very hen or egg question again, right? So we might as well boost. We really don't know.
What would be the optimal time to wait if you had a natural infection? If you had the choice, I know we don't, but what would be an optimal time to wait until between the end of your clinical symptoms and getting the vaccine, do you think?
I mean, I think it's a question how urgently you really need to get vaccinated, right? I think you would have natural infection, let's say, at least for 6 months, what we believe, right? So I guess it's not a bad idea to get a boost at some point, but I think it doesn't need to be urgently, like after 2 to 3 months. So,
do you think in a perfect world, the data that you have on memory B cells at different time points might influence when you'd want to get that boost? Would some of these different sort of memory B cells respond better or worse to that boost?
I think all our discussions right now are so multi-layered. We have public health arguments, we have scientific arguments, we have it is a fascinating question, right? Of course, and everything's moving so fast. Also, it's these are intense times. So they are,
especially with the variants and potential for these antibody escape mutations, and so then. That's your next Nature paper, Christian, is you have to figure that out.
Yeah, we have our hands full with others. I mean, one other interesting thing that came out of this whole study was that with the initial group of people, we were able also to identify neutralizing antibodies, and 2 of them we actually over the summer brought through preclinical models, and by the end of the year, we had the IND enabling studies, and we filed for an IND with the FDA. So we're also, since early January, we started a phase 1 clinical trial at Rockefeller as well to test for safety and pharmacokinetic of those. So we've been kind of juggling these things as well, but these are also, this is very, very, especially for me, when I'm not burned out, I feel extremely privileged, because this is really like the dream of, I think, everyone, every physician especially, right, to be able to go from April 1st, where we had our first participant walk into the hospital, to isolate those antibodies, go through the preclinical testing, and I mean, in those incredible short timeframes that no one thought would be possible, and then really see individuals getting those antibodies and having the potential that they might reach patient care. And the way we're testing those is right now as a subcutaneous administration, because I think what we see for some of those antibody cocktails, they do have the main bottlenecks are administration in a hospital system that is at capacity. So maybe to be able to have an easier access through subcutaneous administration. And so we're right now testing those in our phase 1 as well. So we're really we were able to kind of go full circle, which is really a privilege for me.
So can I ask you a question about that? What patient population would you target for that? Because when I think about antibody immunotherapy for viral infection, you have to catch them pretty early, right? And is there a concern that if it works, that they will then not be protected because they didn't develop their own immune response? Or could enough virus get through to trigger the immune response and they would still develop their own immunity to the infection?
I'm sorry to interrupt. I will have to hop off for another meeting, but I want to thank you all, and thanks, Christian, and your whole team. I know it's it's a lot of people working on it. So great job, and look forward to your other work.
Thank you.
Okay. Bye, all.
Steph.
Yeah, take care.
Yeah, so that's a good question. I mean, I think we would target more or less the same group of people. That's the EUAs for the other antibody treatments are out right now. So symptomatic infections, but early on, so or not severe, so mild to moderate, I think, symptomatic infection, to try to prevent and keep people from the hospital. Mm-hmm. Of course, I think that's an interesting point. What's gonna happen if you give convalescent plasma, if you give monoclonal antibody therapy? Do you keep people from getting there to building immunity due to natural infection? But I think for this specific group, it would rather be to try to keep them out of the hospital and then worry about these things. And then you could probably give the vaccine later on. And but that's another science question, actually, right? How does the T-cell, B-cell immunity look like after you had some treatment here?
So nobody's looked at that, right? Nobody's taken people on given monoclonals and who then to see if they have their own antibodies after those go away. Nobody has done that, right?
I believe there is one study that looks right now into vaccine efficacy after people got some convalescent plasma or a monoclonal antibody treatment. Not sure about natural infection.
I just know that they're recommending if you had that kind of therapy that you wait 3 months before you get the vaccine, I guess, to make sure that it's fully cleared.
Right.
Yeah.
But again, I'm really not sure if people can answer this with certainty, if that would actually even inhibit efficacy. I'm not sure. It might even boost it. Another science question.
I mean, the only analogy we have, and now Steph is gone, but is maternal antibodies, right? If you're getting an infant is getting maternal antibodies, it can interfere with the infant's ability to make their own immune response to an infection.
So, at the risk of changing the subject, since we're talking about antibodies, you mentioned that you're working on a clinical trial of broadly neutralizing HIV antibodies, right?
Mm-hmm.
Is that ongoing, and do we know anything yet?
Yeah, so we're this has been ongoing for the last 10 years in Michelle's lab. So after the identification of broadly neutralizing antibodies against HIV, basically what has happened for COVID over the last 10 months, that happened over the last 10 years. So basically going through animal models in a humanized mouse model, into non-human primates, into the first clinical trials around 2015. And then since I think 2017, a combination of two antibodies has been tested in people living with HIV. And so what we know is that broadly neutralizing antibodies work very well at suppressing viremia. They work well at suppressing active viremia. They also work well in individuals who stop ART. It also delays viral rebound when the antibodies are present. And so I think now a couple of more questions are, do they alter the HIV-latent reservoir? Do they have an effect on that? But those studies are ongoing. And so we're right now building up on a trial that had individuals who got 3 antibody infusions of the combination of 2 such broadly neutralizing antibodies. And then follow them without them taking ART. And we're now having a study that gave 7 antibody infusions and then followed those people living with HIV to study those questions.
Are these intravenous infusions?
So in this particular trial, those are intravenous infusions, but we're also testing subcutaneous administrations for those antibodies to make them also more widely available, because I think that's another important point. And we're also testing those as half-life modified antibodies, so to increase the half-life of those that potentially could reach several months.
Are you ever going to see if they prevent infection?
So we have some studies planned. I mean, with collaborators also in Sub-Saharan Africa to see those, but they're in the planning phase. I mean, right now with the last year, everything was pushed back a little bit, but
yeah, because that would be exciting if in an at-risk population it would prevent, even if you have to give it every 3 months. That's right. That could be good.
Absolutely.
It could also be useful maybe for a post-exposure prophylaxis type of thing.
Yeah. So within 2 weeks you need to do that, right? After infection or exposure. So tell me about the subcutaneous inoculation. My understanding is the volume is quite large, both the HIV and the SARS-CoV-2 monoclonal. So do you put a lesser volume in, and why subcutaneous?
Yeah, that's true. I mean, that's also the question on how much can you concentrate the antibodies, right? What kind of concentrations do you want to reach? In our case, then it also depends on, I guess, activity levels of the antibodies. So what are the neutralizing potencies? If you have higher potency, you can maybe go a little bit less. Obviously, when you think of Regeneron concentrations that the combination was given, some of them were tested at up to 8 grams, right? So
yeah.
Wow.
you definitely don't want to give 100 mLs of product subcutaneously. That does not work, right? So you really have to take those into account. I mean, I think when those antibodies that we were able to isolate, they ranged in the very higher potent levels compared to other monoclonals that have been studied. So I think we're able to keep this balance, to have the volume that we need for subcutaneous administration, but to be able to reach concentrations that we would think are protective. And subcutaneous administrations, I mean, just the route of administration, even when you compare it with IM, would just make things much, much easier. Anything
else, folks, before we wrap up?
I mean, I could ask a million questions.
Yeah, I was thinking the same thing.
But I think we're good.
All right. That's Immune number 42. You can find the show notes at microbe.tv/immune. Send us your questions and comments. One of these days, we'll get back to email, immune@microbe.tv. We have a huge backlog.
Yeah,
and if you like what we do, consider supporting us. Microbe.tv/contribute. Our guest today from Rockefeller University, Christian Gabler. Thanks so much for joining us. Fascinating work.
Thank you very much.
Yeah, congratulations.
This is awesome.
After your next Nature paper, you should come back. That's great. From Cornell University, and on Twitter, Cindy Leifer. Cindy Leifer. Thank you, Cindy.
Thank you.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.ws. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.